We're gonna look at the warm-up, and let me go there. Share screen. Alrighty, so skeletal muscle, like your biceps or your trapezius or deltoid, like all the muscles you normally think about. When they want to generate more or less overall contractile force, we talked about the only way to do it is by activating more or less motor units, which are going to turn on more or less muscle fibers simultaneously. We're going to see that's different when we look at cardiac muscle in just a few moments. Cardiac muscle can actually have more or less contractile force with the same fiber, depending on the presence of hormones and things like that. So all that are true regarding white fibers as compared to red. Remember, white was the fast twitch, also known as the glycolytic. So they use ATP using anaerobic, or they make ATP using the anaerobic pathways. They were like the phosphagen and glycolysis. They contract more quickly. They have actually a faster version of myosin in there that snaps quicker. They cannot be triggered by hormones. That's the smooth muscle that can do that. They contract with more force. What is actually responsible for a white muscle fiber being able to generate more force? What is it about, just anatomically, about a white muscle fiber that allows it to generate more contractile force?
More myofibrils?
Yeah, exactly, more myofibrils. So more actin and myosin arranged in sarcomeres all pulling at the same time. That's also why the white fibers tend to, are larger in diameter because they're just packed with more myofibrils inside. Again, that's when your muscles get stronger, when you're working out and your muscles are starting to bulk out. It's because you're adding myofibrils within the individual muscle cells. So each cell has the ability to generate more force. Smooth muscle, as opposed to skeletal muscle, typically involuntary. Everybody got that? And it generates the force much more slowly. We said like 30 times slower. But the thing that it has going for it is it can sustain a contraction with much less ATP, using only like 1% of the ATP that skeletal muscle would need to sustain a contraction. Does smooth muscle have sarcomeres?
No.
No, it doesn't. Sarcomeres were that really regular alternating arrangement of the actin and myosin that made up the skeletal muscle. In smooth muscle, you've got the actin and myosin arranged more in that lattice, which is why it doesn't look striped under the microscope. Muscle cells stimulated into tetanus, maximum sustained contraction. Again, most people got this. It experiences what we call fatigue. Eventually, it will stop generating force, and it needs to recover. And everybody got this. Word for muscle originally comes from the word for mouse, little mouse. And there was a bit of a clue, like when you started, there was like some little field mice on some poppy heads there. Okie dokie. Stop share. All right, so we're gonna continue now, and we're gonna bring in the last muscle tissue, which we haven't talked about as kind of our segue to get into the cardiovascular system. So what we're going to do now is look at cardiac muscle. And let's do that. And the properties of cardiac muscle that we're going to talk about now will actually be important for understanding some of the properties of the heart. When we start getting into the electrical conduction system in the heart, or we start talking about the Frank Starling law of the heart, or this and that, or things that affect stroke volume and ultimate cardiac output, it's all going to come down to properties of cardiac muscle itself and how they respond to different conditions in your body. So, cardiac muscle. Anatomically, they are single cells, kind of like we saw in smooth muscle. And they also are connected by gap junctions. So in between, if this is these are cardiac muscle cells, they have these things called intercalated discs. And those actually have two of the different kinds of cell-to-cell attachments that we've talked about. One is they have desmosomes. Again, what do desmosomes accomplish? Anyone remember the function of desmosomes as a cell-to-cell attachment?
Resist stretching?
Or more resist. It's kind of resist yanking apart. That was the one that was like the rivets and cable things. So when these are pulling against each other, it's going to keep them from ripping apart from each other. So the desmosomes are basically like riveting these things together. Cardiac muscle does pull in a more linear fashion. It actually has the actin and the myosin arranged more in the that linear fashion. It's actually striated, striped under the microscope, just like skeletal muscle. Even though it's only single nucleus and it's got a lot of other things that are different from skeletal muscle, it is stripy like skeletal muscle, and it pulls, the force is generated along that axis, or as perpendicular to the axis of the stripes. So desmosomes make sure as these things are pulling, they don't pull apart from each other. You don't want your ventricles ripping apart when your heart's beating. And then the other big thing in the intercalated discs are gap junctions. And again, what are gap junctions going to accomplish?
They allow them to be electrically connected.
Exactly. So it means if you stimulate one cardiac muscle cell, that excitation kind of spreads to the whole group that are all interconnected. When we look at the heart in more detail, we'll see that there is a kind of insulating layer of connective tissue that isolates the atria from the ventricles. So if you stimulate the atria, then that excitation spreads across the atria to all the atrial muscle, but doesn't automatically spread to the ventricles without help. Kind of the same thing with the ventricles. Once you start exciting the ventricles, that excitation will spread across all the cardiac muscle in the ventricles. Although, as we're going to see, it gets some help through like the Purkinje cells and the bundle of His and things like that. Other things about cardiac muscle cells that are worth knowing. They can have graded contraction strength. which means that you can stimulate one of these, and how strong it pulls, how much contraction, how much force it generates can depend on different conditions. Like for instance, increased contraction force if epinephrine around. Like the epinephrine will bind to receptors and will actually make these, I think it changes these calcium channels that allow more calcium to come into the cell when you excite it. So you excite it, and the same excitation results in a greater force than you would have had if there wasn't the epinephrine floating around in the body. Right? So this idea that like a twitch of cardiac muscle, it might be a stronger or less strong, depending on whether or not there's epinephrine in the body. Again, this is going to be important a little later when we look at things that can kind of increase cardiac output. Like if you have adrenaline, epinephrine in your body, then your cardiac muscle is actually going to be squeezing harder and pushing more blood out. Another thing that's just like we saw with smooth muscle, well, actually, no, I shouldn't say it, just like, so increased contractile force if pre-stretched. So if it is more stretched out when you contract it, it will push back even harder. This is going to be at the core of what we call the Frank-Starling law of the heart, where the heart can empty out all the blood that's returned to it. If you fill the ventricles even more and they're more stretched out, that next beat is going to squeeze even harder to make sure it pushes all the blood out that it got returned to it. So it's important. This property of cardiac muscle comes up when we talk about the Frank-Starling law of the heart, the ability of the heart to pump out all the blood that's being returned to it. Again, if it's stretched out more to start with, it'll push even harder on the next beat. Other things that are interesting about cardiac muscle. Cardiac muscle has these extended plateaus. Like we normally think of an action potential, whether it's on an axon or whether it's on a skeletal muscle. It kind of goes up and down, and it lasts like a millisecond or something. The action potential on a cardiac muscle cell when it's excited is very extended. It has this like 200 millisecond plateau. This is the action potential here when you're exciting a cardiac muscle cell. This basically prevents summation, right? You can't get tetanus with a cardiac muscle cell. You can't just like excite it a few times really in quick succession and have it just squeeze and not let go. Again, which would be a disaster in your heart. So this kind of prevents tetanus here. What other things do I want to say about cardiac muscle cells? One last real important thing about cardiac muscle cell is that it is autorhythmic. So autorhythmic means you don't necessarily even have to give it a stimulation in order to have it contract. It can just stimulate itself. What happens is it's like this. Like if this is V sub M, and this like this be minus 55 millivolts. So this is kind of our threshold that we normally think about. There are these leak channels in cardiac muscle where the membrane voltage just starts drifting up. When it hits that threshold, boom, you get that extended action potential. Remember, we said it's like about 200 milliseconds across. Comes down, but then it starts drifting up again. And then it hits threshold and it gets an action potential, comes down, but then it starts drifting up again. So we call this like the pacemaker potential. It just goes up and then it drops back. So this thing is happening automatically. You can walk away from it and it's just going to go just on its own. You don't have to make this happen. It's just intrinsic to the muscle tissue itself. I should say that the frequency, how fast this is happening, it depends on the muscle or what part of the heart you're in. But for most of the heart, either the atria or the ventricles, if you cut out a little chunk of the muscle, it would do this, but it would do it pretty slow, like, I think 10 times a second or something. In reality, the autorhythmicity of your heart is going to be driven by what we call the sinoatrial node, or the pacemaker, which is the thing that's going to actually entrain all the rest of the cardiac muscle. That's one of the things we're going to be looking at in our lab today. It's the main thing we're looking at in our lab today, is how all of this cardiac muscle is ultimately going to be autorhythmic, but all kind of slaved into following this pacemaker node up in the wall of the right atrium. So, but this is important. Autorhythmicity, like your heart in general, if you take a muscle out of the body typically and it's sitting on the table, it's just going to go wow. But if you grab, some zombie movie and, drive your hand into somebody's chest and rip their heart out, the heart will just keep beating in your hand until ultimately the tissue dies. But the heart does not need to be connected to the rest of your body or to your nervous system or anything to keep beating. Okie dokie. Right, so with that kind of transition from the muscles to the cardiovascular, we're going to officially introduce the cardiovascular system. Cardiovascular system. Cardiovascular system. So what are these main functions of the cardiovascular system? What does it do for you?
Nutrients and waste.
So one thing it's going to do is going to be transporting nutrients and wastes. Because one of the things I mentioned before, I'll mention again, is, to be a big animal like we are, you are really limited by diffusion, right? If you are just waiting for oxygen to diffuse, we saw in our lab that diffusion is really slow when you get over long distances. Like if we're looking at Mr. Amoeba here, an amoeba or a paramecium or whatever, if you're looking at some little one-celled animal, it doesn't need a cardiovascular system because it's small enough that oxygen can just diffuse into the cell and get to where it needs to go. But when you are a larger animal like we are, then it doesn't work. Like if you waited, like I think somebody calculated, it takes like months. If we waited for oxygen to go like 10 centimeters or something, it would take months. That's not going to work. So instead, we are partitioned into lots and lots of itsy bitsy cells to make up the big animal. And then you've got a crazy number of blood vessels bringing the blood really close within a fraction of a millimeter of every cell. So then the oxygen can diffuse in a reasonable amount of time to get to the cell. Waste can diffuse out and get carried away. So this is from the time you are even just in the womb as a ball of cells, as a little embryo, you've already got your cardiovascular system starting to develop, because you need to be able to start, supplying nutrients and oxygen and getting rid of wastes as you start becoming more than just like a little, blastula or whatever. We're not going to get so into that in this class, but the development of the heart is really trippy because the heart has to continue developing from this really primordial thing pushing blood around in the little embryo to like kind of the adult version of the heart, all the while continuing to beat and do its job while it's turning into an actual heart. So transport nutrients, wastes. What else does it transport actually?
Oxygen.
Oxygen, and CO2. What other things does it do?
Glucose?
That would be like a nutrient. I would say a nutrient. The messaging molecules like hormones. It's the way you transport hormones. We're going to see that, we've talked about, the neurons and releasing neurotransmitters and stuff to send messages. But another really common way to send messages in the body is through hormones, through signaling molecules released into the bloodstream, which then are carried to some target place. We saw one example of that with the epinephrine released by the sympathetic nervous system, which then goes all over. But by the end of the semester, we'll be seeing all sorts of hormones, and not just like the classic hormones like adrenaline or growth hormone, but a lot of organs like your digestive system. Your small intestine talks to your stomach through hormones. Your small intestine talks to your pancreas and your gallbladder through hormones. Your heart can send out messages actually through hormones saying that it's like, we need more blood pressure. There's all sorts of non-traditional endocrine organs that are using hormones to talk and send messages. So hormones are definitely much more than just, glands releasing the classic hormones you've heard of. So we'll see those more. Cardiovascular system's also kind of a staging area for the immune system. You have a lot of the white blood cells are floating around, and when they sense that there's some problem, there's usually chemical signals. They'll actually grab on and leave the closed circulatory system and get out into the tissue spaces and do their magic. That being said, a lot, most of your white blood cells are not in your cardiovascular system. They're actually in your lymphatics and your lymph nodes and just, traveling around in your tissue spaces and the connective tissue and all that. So, we talked about that really briefly last week, but white blood cells are found also all over the place. They're not just in the cardiovascular system.
Also protection, like clotting when there's a cut or something.
Yeah, so we'll talk about that. I don't know if that I'd say is kind of part of the internal maintenance for the cardiovascular system, but we will definitely talk about hemostasis, kind of the whole system to make sure that any kind of break in the blood vessel gets stopped up and healed so you don't bleed out internally. That's going to be an important thing we're going to look at. Again, we got introduced to that idea in the lab where we saw that fibrin being formed in the blood after the blood is leaving the vessel. That fibrin is that's blood clotting. That's part of the kind of patch that the blood makes to kind of seal the hole in the wall of the vessel. Cardiovascular system is also, there's some temperature control. You can dilate or constrict vessels to allow, change the flow of blood to your skin by an order of 50. So if you're trying to cool off, you'll send more blood to the surface where it can radiate off heat. If you're trying to conserve heat, you close down those surface vessels and keep the blood closer to your core to keep warmer. When I first heard that, I'm like, then why does your nose look red when you're cold or something? What supposedly blood gets trapped in kind of these surface vessels, so it's not like you're getting a lot of blood to the surface when you're really cold. It's like the blood is actually being those surface vessels are being kind of closed off, but kind of residual blood that's trapped in there can kind of look reddish. So cardiovascular system, these are kind of functions. Now what we're going to do is just talk about the main parts and pieces of it. Obviously the blood, which is, where we're going to be sending around to transport all this stuff. The heart, which is going to be the thing that's going to be generating the pressure to keep the blood flowing, and all the vessels that are the conduits for the blood. And we're going to look at each of those in a lot of detail. And we'll also be looking at the bigger picture of kind of the fluid flow, basically the physics of fluid flow, the relationships between pressure and blood flow and resistance to flow and all of the things that affect those and how the body uses those to make sure that your tissues are perfused and everything is happy. Okay, so I'm going to start by just kind of laying out the big picture of the cardiovascular system, which is going to look familiar. It's the same picture you've seen in your introductory biology classes. I know most of you have been in anatomy, so you've actually just done this in way depth. But this is just a quick review. You've got the heart. Again, I'm going to, for this class, let's just, the anatomy, the actual anatomy of the heart is kind of more complicated and it's all kind of doesn't actually look like that. But for the purposes of this class, I'm just going to draw it more simply as a cartoon to make it easier. Important to remember that the heart is actually two pumps that are working in parallel. Every heartbeat, you have the right side and the left side independently pumping out to their own circuits. The heart has these kind of these receiving chambers called the atria. Whoops, I just misspelled that. And then the ventricles. Ventricles are the things that are the main pumps. This is the left side of my heart. This is the right side. The left side of the heart is the side that pushes the blood out to the most of the body. We call the systemic circuit of circulation. So we push the blood out. Ultimately, we have capillaries. The capillaries are the things where you actually have exchange. So capillaries, maybe I should type it using my little typer tool. Right, so capillaries are going to be important because that's where either nutrients can leave the blood into the tissue to supply the cells, or where waste can come in. Or so capillary bed. Any vessel that leaves the heart is called an artery. The convention is if the artery contains oxygenated blood, then we draw it in red. When hemoglobin binds oxygen, it turns bright red. So anytime we are trying to show that a vessel has oxygenated blood, we'll draw it red. I'll just mention the main artery that leaves the left ventricle is the aorta. That is like that kind of garden hose, basically, that's leaving the left ventricle out to the body and supplying most of your body cells. After the blood has been in the tissue, it's given up some of the oxygen. It's not as bright red anymore. We're going to find out eventually it actually is coming back to the heart with quite a bit of oxygen still. So it's not like it actually gave up all its oxygen and turned blue, but it gave up some of its oxygen, and it's a little darker, and through your skin, it looks kind of blue. It doesn't actually turn blue. It's more of a bricky red instead of a bright red. And this is going to come back into the other side of the heart. So anytime we have a vessel where the blood is returning to the heart, this is called a vein. And again, that same convention, anytime we draw a vessel where it's looking blue, we're meaning that it has deoxygenated blood. The blood has given up oxygen and it's not fully saturated with oxygen. As we kind of segue into the respiratory system, we're going to talk more about oxygen and hemoglobin and how they bind and things that affect the affinity of hemoglobin for oxygen and all that kind of stuff. But for right now, just think about hemoglobin picking up oxygen and delivering it and then coming back to the heart. So this thing that I've just drawn, this is what we call the systemic circuit. It's going out basically to all your body systems. And then obviously, once the blood comes back to the heart, where does it need to go?
Lungs.
The lungs.
It needs to go to the lungs to pick up some new oxygen. So now we have this thing, the thing to remember here as we leave the heart, whenever we leave the heart, it's going to be a artery. So even though this is blue, this is my pulmonary artery. Which is going to go to the lungs. It's going to be capillaries in the lungs where we're going to pick up oxygen and come back into the left atrium. And again, similarly, even though I'm coming back and it's red. Since I'm coming back, this is going to be my pulmonary vein. So just a lot of times people get stuck on this idea of veins being blue, but veins are only blue on the systemic side. This is going to be my pulmonary vein here. And it's red. And so this side here, I'm going to call my pulmonary circuit. Pulmonary meaning having to do with the lungs. So this is two of the main circuits here. Another circuit that's important to keep track of is the coronary circulation. Like the heart actually uses a lot of energy to keep pumping your blood, and it's important that it gets the first dibs on the supply, right? It's kind of like if you're on the airplane and the masks fall down, first thing you do is put the mask on yourself so you don't pass out, so you can take care of like the people you need to take care of. So the heart actually gets blood directly off of the aorta. They call them the coronary arteries. There's a right and left coronary artery that basically go directly into the cardiac muscle tissue and supply the cardiac muscle itself. Again, those coronary arteries arise right at the very base of the aorta there. So that's my coronary circulation, making sure that the heart has plenty of blood. And the cardiac muscle is basically purely aerobic. So it needs a good supply of oxygen to keep beating well. These are some of the, like when we talk about control of blood flow later, in general, your body will adjust the amount of blood that goes to different parts of your body depending on the tissue's needs and this and that. The coronary arteries basically are always full bore. You don't, if your body's trying to conserve blood because you're bleeding or something, you're not going to constrict the coronary arteries because ultimately it's not going to buy you anything. If your heart dies, the rest of your body's up a creek anyway. So you do your best to make sure that the heart gets what it needs to keep doing its job. What do we call it if something blocks the coronary arteries and the heart muscle is becoming ischemic, isn't getting blood flow?
Heart attack?
That's a heart attack. Heart attack is when there's a blockage of these coronary arteries and the heart muscle is not getting its blood. There's different names for it. You can call, so heart attacks. It sounds like it's going at you. Oh my God, the heart's attacking. But no, it's, so there's different, you can have, like we talked about strokes, it's a similar kind of model, right? You can have a temporary blockage of the coronary arteries, coronary blood flow. You can have a TIA, a transient ischemic attack, in which case you get what's called angina pectoris, which is kind of crushing pain, but ultimately it passes. So this is a temporary transient ischemic attack. Again, right, ischemia is lack of blood flow. If you end up having the blood flow blocked for longer, then you start getting the cardiac muscle dying and being damaged. And then we get like, MI, myocardial infarction. Myo is muscle. So myocardium is the heart muscle, cardiac muscle, and infarction means there's actually tissue damage. So this would be kind of the worst, kind of more the result of a more serious heart attack. And you can get this either because there is some errant blood clot, like that fibrin has broken loose and got stuck and clogged up a vessel, or it can be more long-term, this buildup of these little atherosclerotic plaques that we'll talk about, where the blood vessels end up getting so restricted because of the stuff in the walls that the blood flow doesn't, that the blood isn't flowing well anymore. So heart attack, this is all about blockage of the coronary arteries and the cardiac muscle not being able to get its supply of blood it needs to be healthy.
What role does stress have in this, if at all?
I think stress increases your chance of getting like those atherosclerotic plaques is partly like kind of, having stress, you have more like inflammatory responses and things. But I don't know so much. I don't know. Maybe somebody's going to cover that in their presentation at the end of the semester. Definitely diet can affect the deposition of the atherosclerotic plaques to impede blood flow. I think stress also plays into some of those same mechanisms. So these are three of the circuits I've seen so far. We have our systemic circuit to the body, pulmonary circuit to the lungs, coronary circulation supplying the cardiac muscle itself. The one other kind of circuit I want to mention is a portal system. We're going to see a few different portal systems as we go through the semester, but I'll introduce the first one right now, the hepatic portal system. So in portal systems, rather than that kind of out and back, the basic idea of portal systems is you have two capillary beds. Basically the first capillary bed drains into a second capillary bed that then ultimately returns, drains back to the heart. So kind of an example of this. The hepatic portal system. So here's our heart. And we talked about, sending the blood out to the body. Returning back to the other side of the heart here. So this is the systemic circulation here, where the blood's coming out and it's coming back. Again, that's just the normal. There's one capillary bed. An example of the hepatic portal system, basically, like the blood that's going out to your digestive organs. So this is like your stomach and your intestines and all that. This is going to be capillary bed number 1. And it does not go straight back to the heart. After you've absorbed all those nutrients and stuff, it goes to the liver, to a second capillary bed in the liver, where those nutrients can be processed, or toxic things can get detoxified, or things can get stored for later. So this goes into capillary bed number 2 in the liver. And finally, those drain back into the normal, to the heart. But there's two capillary beds. This thing right here, we call this the portal vein. Right, the portal vein does not drain back to the heart. It drains into capillary bed number 2. So when we talk about the hepatic vessels, there'll be like the hepatic arteries that lead into, blah, blah, blah. These are the mesenteric arteries that lead into the digestive organs. But then the digestive organs drain into this hepatic portal vein, which goes to the liver, and then this hepatic vein, which then drains the liver. But the hepatic portal vein, even though we call it a vein, is actually entering the liver. So hepatic portal vein enters to the liver, whereas the hepatic vein leaves the liver to then go back to the heart. So does this make sense? This is because when you absorb stuff from the digestive tract, you want to be able to process it. The liver, we'll talk more about the liver. It's doing all sorts of stuff to detoxify, to transform, to store, all this and that. We'll see a portal system in the when we get more into the endocrine system, the connection between the hypothalamus and the pituitary gland, which we've already talked about a little bit. When we look at that in more detail, we actually have a hypophyseal portal system between the hypothalamus and the pituitary gland. When we get to the kidneys later, we're going to see a portal system there where you have the glomeruli, which are these first leaky capillaries, which then feed out their efferent arterioles into the peritubular capillaries. So we'll see different versions of this, but for right now, let's just stick to this idea of the hepatic portal system.
Can you say again kind of the end of this where you said the hepatic vein, which one of them drains the liver, which one of them enters the liver, and then which one drains? I was confused about that.
So the portal vein is the one that's entering into the liver. This is the liver here. And the hepatic vein, so yeah, it gets confusing, especially when you're in anatomy, because you have the hepatic, it's called the hepatic portal vein, and it actually enters the liver, and then you have the hepatic vein, which leaves the liver. So you have to really make sure you keep track of that word portal, because hepatic portal vein is the thing draining the digestive organs, entering into the liver. The hepatic vein is the thing that's ultimately leaving the liver to go back to the heart.
Okay, got it. Thanks.
Okay. So that's kind of giving you a sense of these main circuits. We are going to go back to this picture here now. Things to kind of keep track of here, to make sure that the blood keeps going in one way and doesn't like back up. We have little valves. You should be aware of the valves. There are valves between the atria and the ventricles, or atrioventricular valves. Right, so when the ventricles are squeezing, the blood can't squish back up into the atrium. So the AV valves are important because they keep the blood from squishing back up where it came from. When the ventricles are squeezing hard, the blood will have to squeeze out to the arteries leaving the heart, either the aorta or the pulmonary trunk. The one on the right is the tricuspid. The one on the left is the bicuspid, also called the mitral valve. but they're both basically one-way valves going, allowing blood to come in from the atria, but not go back up when the ventricle's squeezing. And then once you push the blood out into the system, there's a lot of blood pressure now, right? You're 120/80, which we're going to look at in more detail later this week. There's a lot of blood pressure here. You don't want the blood coming back into the heart while the ventricles are refilling, getting ready for the next heartbeat. So we have the other valves that are important are right here at the base, at the beginning of these major arteries leaving the heart. Those are called the semilunar valves. So when the ventricles, we're going to talk about this word more, diastole. When the ventricles are in diastole, when they're relaxed and refilling, this is going to make sure that all the residual blood pressure out in the system doesn't squish back up into the heart. It stays out in the vessels where it was pushed out into. So the semilunar valves are the one-way valves that keep the blood from squishing back up into the heart from once it's been pushed out into the arteries. When you hear the heartbeat, this is going to be another thing we're going to be doing later on in lab as well. Every time the heart beats, you hear the sound of the valve slamming shut. When the ventricles start to squeeze and they start building up pressure, the first sound you're going to hear are these AV valves slamming shut, right? So the ventricles start squeezing, the AV valves slam shut to make sure the blood doesn't squish back up into the atria. That's that lup sound. And you're pushing blood out into the body. And then when the heart relaxes, begins diastole, then now these semilunar valves slam shut to make sure the blood doesn't return back up into the ventricles as they relax and refill. That's the second sound, the dup sound. Like lup dup, lup dup, lup dup. That's going to be AV valve slamming shut as the ventricles squeeze or in systole, and then dup as the ventricles relax, go into diastole to make sure that the blood doesn't backflow from the arteries. So we'll be talking about these in more detail, but you should definitely be aware of them, making sure that the blood continues to flow in one direction. So if we look at these two circuits, it's important to realize that the flow in each side has to be balanced. So when we talk about the blood flow, I'm going to introduce this idea, cardiac output. Cardiac output. This is the flow, liters per minute. From either ventricle of the heart. And you might say either ventricle. Why shouldn't it's maybe it's going to be different. You don't have to, there's the cardiac, the circuit going to the lungs is so much smaller than the circuit going out to everything else in your body. But the flow, the cardiac output has to be balanced on both sides, or else things will back up and get back pressure. So I want to make sure this makes sense to you, because basically everything you push out of the left ventricle that goes to the systemic side is returning to the right side. Everything you push out the right ventricle to the lungs is coming back and returning to the left atrium on the left side. So with every heartbeat, everything that pushes out the right side is arriving at the left side. And everything that's pushing out at the left side is coming back and arriving on the entrance to the right side. So if there's any imbalance on one or the other of these ventricles pushing out, it's going to result in the back pressure building up on one side or the other side, either the systemic or pulmonary circuits. So the cardiac output has to be balanced. So cardiac output is the same on the right and the left. Does that make sense? If one side of the heart is able to pump better than the other side, then it's called congestive heart failure. Congestive heart failure is when the heart is not able to pump efficiently and one side starts building up back pressure because one side is pushing more flow than the other. In that case, there's two possibilities. If the left side is pumping better than the right side and the right side can't keep up, you're going to build up pressure on the systemic side. That's called peripheral congestion. Let me. Again, if the left side is working harder, you can get peripheral congestion. If the right side is working harder and the pressure is building up in the pulmonary circuit, you can get pulmonary congestion. These are going to lead to like edema. Like if you have too much pressure in the vessels, you're going to have fluid leaking out of the vessels into the tissues. So it's not a good thing. You want to make sure that the cardiac output is balanced on both sides. That doesn't mean that the pressure is going to be balanced. We're going to see in a few moments, you need way more pressure to actually keep the blood flowing in the systemic circuit because there's so much more blood vessels. So much more resistance because of all of the length of the vessels to get to the systemic circuit. But the cardiac output, the amount of flow on the right and left has to be balanced. So keep that in mind as we keep continuing on. This picture is ugly, but I think I've kind of laid it out in a nice way. So if you watched it from the beginning, you don't just use this picture as a study guide because it looks like vomit. All right. What I'm going to do now, we've kind of laid out these big picture things of just how the kind of cardiovascular system is laid out. But what we need to do now is get into the details of more the physiology, which basically is the physics of fluid flow. Luckily, it's all pretty straightforward, and I feel pretty intuitive. And also, it's going to be the same basic relationships that we saw with like current, electrical flow. It's the same relationships that we are going to see when we look at the flow of air when we get to the respiratory system. But let's first introduce the idea of flow. So blood flow. This is going to be liters per minute. How much volume is moving in any particular amount of time? It's important to realize that flow is not the same as velocity. So like for instance, I might have a vessel really big and the blood is just kind of gloop, going pretty slow, gloop, 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 gloop. But it might fill like 1 liter in 1 minute. I might have a smaller vessel where the blood is really having to come really fast, but as it fills up some bucket here, it's still, 1 liter per minute. This blood here coming out the small little thing is going way faster, but the flow is exactly the same. This is how you end up like kind of soaking your friend on a hot day in the yard. You put your finger over the garden hose nozzle. The same flow is coming out, but in order for that same amount, that same liters per minute to get out of the small aperture, because your thumb is blocking most of it, the water has to come out way faster and travels much farther before gravity pulls it to the ground. Right? So I just want to make sure people get that flow is liters per minute, how much liters per minute, but it's not really about how fast it's going. You can have the same flow at high velocity and low velocity, particularly in this example of something with a larger diameter and smaller diameter. To keep the same amount of blood moving through a small pipe, it's got to be going faster to get through. So flow. When we're talking about the flow in the entire system, we're going to be talking about cardiac output. Right, this is the flow from either ventricle. Cardiac output, also called CO. We'll also be looking at local flow, right? Sometimes we'll be looking at flow in a more, without looking at the big picture, just thinking of flow as just flow. But when we're talking about some of the control of your overall blood pressure in your body and flow, the flow is going to be important. We'll be talking about the cardiac output. So now let's talk about pressure. What makes the flow move? The movement happens because of pressure differences. There's some delta pressure. If you have a higher pressure on one side versus the other side, there's going to be some flow. So the higher the pressure, the greater the flow. The lower the pressure, the less the flow. It's the same thing like voltage. Voltage was the pressure, so to speak, when we did the electrical stuff. Higher the voltage, the higher the electrical current. Here, the higher the blood pressure, the greater the blood flow. Then the other thing that's going to be affecting things is resistance to flow. Resistance can come for a lot of different reasons, which we're going to talk about in just a few moments. But basically, resistance to flow is saying for a given amount of pressure, like how easy is it for the blood to actually flow, right? If you have a lot of resistance to flow, you're going to need more pressure to get the blood move, the same amount of blood through. If you have very low resistance, you're not going to need as much pressure to keep the blood moving. So the relationship between pressure, resistance, and flow, these are the three main things that we are going to be looking at in detail for the next few weeks. There's a really simple mathematical relationship that you need to know really well. You got to really just kind of feel super comfortable about it. I think it's pretty intuitive. There's two main ways you can describe it. You can say, this is one way that I think is, flow is going to be pressure divided by resistance. Right? If you think about this just kind of intuitively, increasing the pressure increasing the flow. Right? If there's more pressure, you're going to have more flow. There's less pressure, there's less flow. So that part makes sense why that's on the top. When we get into the denominator, resistance, this also kind of makes sense. Assuming pressure is staying constant, if the resistance is going up, you're going to have less flow, right? Because it's harder for stuff to make it through. If resistance is going down, the flow is going to go up. If I have increased resistance and I want to keep the flow at the same level, I'm going to have to increase the pressure as well, right? So really kind of think about this relationship and make sure you get it into your head really well, because it's going to be at the core of so much of our discussions of control of pressure and control of different things. We can also rearrange it another way, particularly because I said we're going to be talking about control of blood pressure. Pressure, if we just multiply both sides by resistance, this is going to be flow times resistance. This is the same equation, except now we're like looking at pressure. So as we want to see different ways the body can adjust your blood pressure. It can do it two different ways. It can either change the amount of flow. And again, flow, if we're talking about the big picture, cardiac output, having the heart push more or less blood through, maybe by increasing or decreasing your heart rate, for instance. Resistance to flow is going to be, we're going to see in a few moments, the main way you can do that is by changing the diameter of blood vessels. If blood vessels are constricted, it's going to be more resistance. So if I constrict some major arteries and the resistance goes up, that will actually help bring my blood pressure up. So thinking about this equation and this equation, again, these are just two different versions of the same equation. These are going to be critical for you to kind of make sense of everything we're going to be talking about over the next couple of weeks in terms of how blood pressure is controlled by the body and how you kind of deal with all of this. Are there any questions about these two main equations? Right, so now what I'm going to do, we need to spend a little more time looking at resistance. We'll spend a lot of time talking about pressure and stuff, but resistance is going to be obviously at the core of adjusting blood pressure. So we need to see the things that affect resistance to flow in your cardiovascular system. So what kind of things are going to affect the resistance to flow? I'm looking at just some system, some blood vessel and some blood flowing through it. What kind of things are going to make it harder or easier for that blood to flow with a given amount of pressure?
Plaque buildup.
So in the plaque buildup is ultimately doing what? So plaque buildup is going to affect it. And why? What does the plaque do?
Thickens the walls of the vessel.
So it thickens the walls, which then ultimately, what's that going to do? As the walls get thicker, what's happening?
It constricts the diameter.
Yeah, so that's it. Ultimately, it's changing the diameter of the vessel. So the thing that we really want to focus on is the diameter of the vessel and how the diameter of the vessel affects resistance. And the relationship is crazy. So resistance, which I'm going to do as an R, is proportional. This is just proportional to the inverse of the fourth power of the radius. So it makes sense that resistance and radius are inversely proportional, right? If I have a blood vessel and let's say it has a radius of r, and I have another blood vessel here and has a radius of 1/2 r. And I have another vessel here and it has a radius of 1/3 r. It's pretty obvious just from your intuition that the wider thing is going to have less resistance to flow and the narrower thing is going to have more resistance to flow. It ultimately happens because of kind of surface effects where the blood actually interacts with the wall. Like when you're in the middle of the vessel, there's what's called laminar flow. Things just kind of get in a groove and are just moving along. But wherever the blood is actually touching the walls, you get kind of friction. Where the blood is having to deal with this friction of the wall of the vessel. So the higher proportion of the blood that's just in this flow versus interacting with the walls, the less resistance. When we get a small thing here, we have way more blood dealing with the resistance with the walls and not much in that kind of laminar flow going down the middle. So that's another kind of hopefully intuition why a smaller thing has a lot more resistance. But now let's go back to this relationship. Resistance is proportional to the fourth power of, or inversely proportional to the fourth power of the radius. So let's say the resistance to flow in this case, let's say resistance is R. What is the resistance to flow going to be in this case when we have 1/2 R, half of the radius? Right? 1/r to the fourth. If r is 1/2 little r, that's going to be 1/1/2 little r is going to be basically quantity cubed. That's going to be little r to the it's going to be 2 to the fourth r. What is 2 to the fourth? Oh, come on. What does the fourth power mean?
Is it 64?
So not quite. What does it mean? What does the fourth power mean? What does it mean if I say what does it mean if I say r squared? What does r squared mean?
R times R.
It means r times r. If I say r to the fourth, what does that mean?
R times R times R times R.
Yeah. So definitely exponents are something you need to understand for this class. They come up, and they came up with BMI. They'll come up in other things. You definitely need to know kind of the basic arithmetic stuff for, when we talk about, the basic physics of physiology are not getting into, if you want to get more complicated, you can get into more advanced math. But definitely some of the basics, you need to know kind of basic things like the multiplication, division, exponents and stuff, and feel comfortable. So r times r times r times r. So 2 to the fourth is going to be
16.
exactly. It's going to be 16 times r. So even though it's just half the diameter, it's got 16 times the resistance here. If we look at this case, this is going to be basically 3 to the 4th times R. What is 3 to the 4th power?
81.
It's going to be 81, right? 3 times 3 times 3 times 3. 3 times this is 9 times 9. It's 81. It's going to be 81 times R, right? So you can see there's this really intense relationship between diameter and resistance to flow. So that smooth muscle in the blood vessels is going to be a critical part of control of your blood pressure, because little changes in resistance, little changes in radius make big changes in resistance. And like I was saying over here, this resistance, by changing resistance up or down, you're adjusting blood pressure. So this is going to be a major part of blood pressure control, is by adjusting the diameter of the vessels. A little bit of vasoconstriction or vasodilation has a big change in resistance, which can change blood pressure. So you're going to see how these pieces all start interweaving with each other. More things about resistance. So one thing is diameter. Maybe I should say radius, because I've just been talking about radius. Again, resistance is proportional to the 4th power of the radius. Here you can also see why those plaques, those buildups in the walls of your vessels, can be so concerning. Because by changing the diameter or the radius, making the passageway narrower, it has a really dramatic effect on how difficult it is for the blood to flow through that vessel. Other things that can affect resistance to flow. Another one that's going to affect, or that we're going to be concerned about, is just the total length of the vessels. Basically in your body, you've got about 60,000 miles of blood vessels, 100,000 kilometers of blood vessels to supply all of the different cells in your body. And that eventually, the more like length of vessels you have to go through, the more resistance ultimately you're going to be generating, the more pressure you're going to need to keep the blood flowing. In fact, if we come back to this train wreck picture here, if we take a look at these two circuits, the systemic circuit and the pulmonary circuit, right? I was really clear saying cardiac output has to be the same on the right and the left. The flow is the same on the right and the left. But the resistance is very different, because to send blood to all of this, in fact, maybe let me use a different picture. This is just looking so ugly. Oh, come on. Hold on. I should do red. So the systemic circuit, coming back to the pulmonary circuit. So the systemic circuit you know, and the reason why the resistance in the systemic circuit is so much larger is just because you have so many more vessels to supply the whole body versus the other side, the pulmonary circuit. It's like I go from the heart to the lungs, which are right there, they're their neighbors, and then come back to the heart and you're done. So there's so much less distance of vessels, so there's much less resistance. So what does that mean? Given that the flow has to be the equal on both sides. So if I'm looking on this side, the flow is equal on both sides. But the resistance is going to be much higher on this side. So how am I going to have, I know flow is pressure over resistance. So if I know that the flow has to be the same, but the resistance is 4 times higher on this side, what do I know about the pressure on the systemic circuit?
That the pressure also has to be higher?
Exactly. If the resistance is 4 times higher, then the pressure's got to be 4 times higher to keep the flow the same. So when we, make sure you don't get pressure and flow confused. Like if there's some question like, how does the flow on the systemic side compare to the pulmonary side? They're the same. How does the pressure on the two sides compare? It's much higher on the systemic side. Most of the time when we talk about someone's blood pressure is 120/80, we're talking about their systemic arterial blood pressure. The blood pressure on the pulmonary circuit is going to be way less than that. And finally, still talking about resistance. Whoop, that's not what I meant to bring up. Oh, where did I do it? Oh, I think I just got rid of it. Okay, so resistance. There was radius to total distance, total length of the vessels. The one last thing I'll put in here, because it can be relevant, it's not going to be relevant for most of our discussions, but you should know that it's another thing, is the viscosity of the blood. Right, this is an intuitive thing if you've ever had a milkshake, right? If you try to suck up milkshake through a straw, it's more work. You have to generate more pressure differences than if you're sucking down a Coke or a Sprite, right? So if the blood is thicker, it requires more pressure to keep it flowing because there's more resistance to flow. For our class, we're not going to be talking about viscosity that much, but you should be aware of it. Like one thing that can affect viscosity is just, hematocrit. We talked about hematocrit, how many blood cells there are compared to the amount of plasma. Particularly if somebody's doing blood doping or something, taking erythropoietin to like unnaturally increase the number of red blood cells in their blood, it can actually get to a point where the blood becomes so thick that resistance to flow becomes a problem in keeping the blood going well, because it becomes more sludgy. You kind of got mud you're trying to pump through your vessels instead of just kind of more fluid blood. I should also mention the viscosity of blood is very nonlinear. When people talk about the study of blood flow and blood viscosity, there's this idea of blood rheology. It's because blood is kind of weird. Those little blood cells that we talked about, they have electrostatic attractions to each other. If the blood really slows down, they actually start assembling in these things are called rouleaux. Kind of like a stack of lifesavers, which makes the blood even more thick because now all the blood cells are interconnecting with each other and making all these kind of stacks. But then once things start flowing and the rouleaux break apart, all of a sudden things are way thinner because now they're not, so this idea of the viscosity of the blood, it's not just about how many blood cells are there, but actually the speed of the blood can affect the viscosity because of this weird nonlinear effect where the blood cells get attracted to each other and assemble into these stacks called rouleaux and all that. So just putting that out there, we're not going to cover that in this class, but just so you know that viscosity of blood and all that is kind of this nonlinear thing that gets more complicated if you do care about it in more detail. So resistance, we'll definitely be spending a lot of time talking about how radius of blood vessels affects resistance and therefore is going to be affecting either blood pressure or flow. Total length of vessels is important, particularly because it explains why the systemic circuit is got such higher blood pressure than the pulmonary circuit. This also explains why being overweight is not good for your heart health, right? If you've got a bunch of extra flesh on your body, that has to be supplied by blood vessels. So by adding, by having, that extra flesh on your body, you're going to be adding, probably thousands of miles of extra blood vessels to your systemic circulation. That extra length of blood vessels means that there's more resistance, which means that your blood pressure has to be higher and your heart has to work harder to keep the flow going. So if you lose a bunch of weight, that's actually taken a bunch of work off of your heart because there's less blood vessels and less overall resistance to flow for your heart to deal with. So this total length of vessels also comes into the reasoning of why being overweight is not necessarily healthy for your heart.